Hello, everyone. It's an honor to be here today to share our research findings with you. Our current research supports the idea that outer membrane vesicles may represent a novel regulatory mechanism for myocardial injury in high-altitude environments. The gut microbiota plays a significant role in various physiological and pathological processes, thereby influencing host health. Outer membrane vesicles, as an important component of their secretions, can facilitate intercellular communication and transport bioactive molecules. In high-altitude environments, outer membrane vesicles can cross the intestinal barrier to affect cardiac function, and they may exert their effects by modulating inflammation, oxidative stress, and apoptosis in cardiomyocytes. Through genetic engineering modifications, the targeting ability and retention time of outer membrane vesicles can be enhanced, thereby improving their therapeutic efficacy. The treatment methods for high-altitude myocardial injury include interventional therapy, drug therapy, and surgery. Interventional therapy encompasses the implantation of cardiac pacemakers and percutaneous transluminal septal myocardial ablation. Drug therapy primarily involves the use of sodium nitroprusside, dobutamine, beta blockers, and calcium antagonist. Sodium nitroprusside is a vasodilator that stimulates the release of nitric oxide from the endothelium. Dobutamine is a drug that targets the beta-1 adrenergic receptors in the heart. Beta blockers can reduce myocardial contractility, relieve coronary artery obstruction, decrease myocardial oxygen consumption, and increase ventricular volume. Calcium antagonist can reduce myocardial contractility and improve diastolic function. Surgery mainly involves the resection of hypertrophic myocardial tissue to reduce the degree of hypertrophy. The gut microbiota plays a crucial role in host health, with outer membrane vesicles facilitating intercellular communication and the transport of bioactive molecules. In high-altitude environments, the gut microbiota can significantly influence cardiac function through outer membrane vesicles secreted by both beneficial and pathogenic bacteria. The first mechanism involves the cross-linking between peptidoglycan and the outer membrane being reduced by the denaturation and hydrolysis of lipoproteins, which triggers the formation of outer membrane vesicles. The second mechanism is the accumulation of periplasmic proteins that exert pressure on the outer membrane, inducing the formation of outer membrane vesicles. The third mechanism involves the deacylation of lipidae in the outer membrane, leading to membrane remodeling and increased curvature, which promotes the formation of outer membrane vesicles. Outer membrane vesicles are commonly extracted from bacterial supernatants using methods such as ultracentrifugation combined with ultrafiltration, immunoaffinity chromatography, and detergent-based extraction of detoxified outer membrane vesicles. The characterization of outer membrane vesicles involves various techniques, including protein electrophoresis, gel electrophoresis, enzyme-linked immunosorbent assay, scanning electron microscope, and particle size analysis. Their functions include applications in vaccine development, drug delivery, and genetic engineering. In this study, we have identified that outer membrane vesicles secreted by the gut microbiota represent a novel target for high-altitude myocardial injury. However, the mechanisms of outer membrane vesicles formation and their pathogenic roles have not yet been fully elucidated. We will continue to investigate the pathogenic targets of outer membrane vesicles in myocardial injury and aim to develop new targeted therapies for myocardial injury based on the drug delivery characteristics of outer membrane vesicles, thereby providing new approaches to improve the health of people living in high-altitude regions.